Replication is essential for the division of cells and is part of the central dogma of genetics. In prokaryotes, replication proceeds as follows. First, the initiator protein binds to the origin of replication. Then, helicase unwinds the double-stranded DNA and gyrase releases torsional stress. Next, the single-strand binding protein binds to the unwound single-stranded DNA to help stabilize it. Shown in light blue, RNA primase adds a short primer in pink that is complementary to the template DNA strand. The primer is necessary to give DNA polymerase a hydroxyl group on which to start adding nucleotides. Starting at the primer, DNA Polymerase III adds nucleotides that complement the template strand. Because DNA synthesis happens from the 5' end to the 3' end, one strand is synthesized towards the replication fork and the other strand is synthesized away from the replication fork. On the template strand that runs 3' to 5', the DNA polymerase continuously synthesizes DNA, catalyzing the phosphodiester bonds between nucleotides. The continuous nature of synthesis makes this DNA replication strand the leading strand. On the other template strand, which runs 5' to 3', lagging strand synthesis takes place. On the lagging strand, DNA is synthesized in short pieces called Okazaki fragments. The lagging strand also has multiple primers instead of just one, since DNA polymerase III has to restart to synthesize the more recently opened part of the template DNA. The multiple primers are destroyed by DNA polymerase I, shown in yellow, which also replaces the primers with the correct corresponding nucleotides. The final step in lagging strand synthesis is to ligate the fragments of DNA together into a single complete strand, which is done by the enzyme ligase.